So a slice of living human brain kept alive in a lab dish? This just changed what we thought we knew about Alzheimer's. Are we actually on the verge of rewriting how we think about genetic diseases?
Right. And just to set the stage, today is May 8th, 2025. Alzheimer's, sickle cell anemia, beta thalassemia. These aren't just names. They've shaped global health for decades. Millions affected, families overwhelmed, healthcare systems stretched thin. Why do these diseases still seem so tough to treat?
Yeah, exactly. For so long, the options for both Alzheimer's and those blood disorders have been, well, really limited. Alzheimer's is now the main cause of dementia worldwide. It takes away memory, independence, and honestly, sometimes even the sense of self. And with sickle cell and beta thalassemia, people's lives are shortened and they're dealing with constant, costly care. But there's finally something big happening.
So let's start with Alzheimer's. Last month, scientists managed to keep slices of living human brain tissue alive in the lab. What exactly does that change?
It's, honestly pretty huge. For the first time, researchers could watch how amyloid beta, the protein we think is central to Alzheimer's, actually damages the links between brain cells in real time. Even small changes in amyloid beta levels messed up how those cells communicated. Before this, we were stuck with animal models or looking at brain tissue after death. Now, it's direct evidence from living human brain slices.
Does that mean, are we actually close to a cure, or is that jumping ahead?
Yeah, it's a bit early to say that. Alzheimer's is complicated. We still don't have the full picture. This new data is a big step, but getting from that to safe treatments is a long road. We'll need a lot of clinical trials, and they have to involve really diverse groups of patients before anything new becomes widely available.
Meanwhile, the genetics side is moving quickly too. There's that new finding about the Casp8 gene mutation, right?
Yes, so this mutation in Casp8 seems to double the risk for late-onset Alzheimer's. What's interesting is, it causes buildup of toxic proteins that aren't amyloid or tau, the ones we usually talk about. Which makes you wonder, are there other risk factors out there we haven't even found yet? Honestly, probably yes. That means prevention and treatment could get a lot more personalized.
Okay, let's switch to gene editing. Casgevy, the first CRISPR therapy, just got approved for sickle cell and beta thalassemia. How does it actually work?
Casgevy edits a person's own stem cells. First, doctors take the stem cells out, then use CRISPR to fix the faulty gene and put the cells back in. The idea is that if it works, it could cure the disease. It's still expensive, but less than half the lifetime cost of usual care. The bigger issue is access. Not every hospital can do this, and patients need close monitoring throughout the whole process.
If it's really that effective, why is it rolling out so slowly?
Yeah, it's mostly about regulation and logistics. Any gene editing treatment has to get through strict safety and effectiveness checks, and only certain medical centers are set up for these complicated procedures. And there's still debate, ethical questions about how and when we should use this kind of technology.
That brings up another issue. With CRISPR in humans, what are the biggest ethical or safety worries?
Well, off-target effects, unintended genetic changes are one worry. There's also the question of equity. Who gets access? And could these treatments actually widen health gaps worldwide? And if we're talking about editing the germline, heritable changes, that's a whole new level. Most countries have very tight rules or outright bans on that.
So even as the science moves forward, there's this question: who benefits first? Wealthier countries have the means, but low and middle income places face real hurdles. What's being done to close that gap?
There are some efforts, like expanding treatment centers and setting up tiered pricing. Some groups are pushing for wider insurance coverage too. But unless lower-income health systems get more resources, the benefits are still going to be uneven.
So, bottom line, these advances bring hope and progress, but access is still a long way off, and the ethical questions are really just beginning. Are we ready for what comes with changing our genetic future?
We're at a crossroads. Living brain tissue research and gene editing bring us closer to actual cures, but they force us to rethink who gets access, how we handle safety, and where responsibility lies. The challenge now is making sure these breakthroughs reach everyone.
Thanks for listening as we dug into the science behind the news and tried to figure out what comes next. Stay curious, and we'll be back next time.